This is the 14th podcast on human molecular genetics, and the overall idea behind this section is about understanding the whole DNA molecule and the human DNA molecule, and then figuring out the sequence of alleles, and then from there being able to use it for, hopefully for the benefit of everybody, to prevent disease and to screen people. Well, the first thing you need to do is you need to actually figure out what alleles are on the human chromosome, and there are many techniques that they use for determining this. A lot of times when parents are going to have a child, they go in for genetic testing, and what they're looking for is the alleles for disorders that may cause disease in a child between, the offspring of two parents. And usually you're testing for the presence of recessive alleles of genetic disorders. And one of the techniques is called a DNA probe, where you take a piece of DNA that's radioactive and it will bind to a sample you've taken from the parents of the DNA, and it will bind to a sequence found in a disease-causing allele, and from there they can detect the presence of a disease. And other techniques that are used are and also used within the DNA probe is restriction enzyme analysis, which we use at the DNA Learning Center, where you cut the DNA with enzymes and use electrophoresis to cause the segments of DNA to move through a gel, and from there you can see the bands of clumps of DNA to fit, and which also can indicate the presence of certain alleles. Now, one of the techniques that uses gel electrophoresis is with DNA fingerprinting, and what's done there is you take DNA, and you're not so much looking for genes as much as you're looking at big chunks of DNA that really don't code for anything. They have little or no known function at the time, and you're going to find that parts of that DNA have repeating sequences, and those repeating sequences can then be used to identify the person. So what they will do is use these segments called repeats, and you can see here that this person has 12 repeats, this person has 9 repeats, so the segments of the DNA are going to be a little bit different. And what they do is they cut the DNA using an enzyme, and you can see this person, the length of this piece right here is pretty long compared to this piece. And then they put the pieces into the tracks of and use gel electrophoresis, and the segments travel down these tracks because they are polar molecules, and from there the smaller pieces will travel faster than the larger pieces, and you start to get these series of bands which help us determine the genome of the person, and this is what they call a DNA fingerprint. And because the DNA is different from person to person, people get different bands, and that's called their DNA fingerprint. So now that they have, we know how to work with the DNA. A project was taken around the world to sequence the entire human genome, every single gene in the human genome. And in 2000, they started in 1990 and they finished in 2000. And now what they're trying to do is analyze these sequences to see if, to look for genes and alleles for disease. And just to give you an idea how big the task this was, there were 6 billion base pairs in the human genome, and if you printed it out, it would fill 1,400 of your biology textbooks. That's an enormous amount of information. So one of the techniques they use is what's called the shotgun sequencing, which is rapid sequencing in your I think in your outline. And what you do with shotgun sequences is you take the DNA and you cut it into fragments, and then what you do is sequence each of the bases of the fragments by determining adenine, thymine, guanine, cytosine, the exact sequence of them. And then you take these little pieces and you start to look for overlapping areas and put the pieces, link the pieces of them back together to finish this sequence. And it's very much like a jigsaw puzzle. It's a very simplified explanation of it, but that's one of the procedures they use. Another thing that scientists are doing now that they have the actual sequences, they start looking for genes. And one of the things they look for is what's called a promoter sequence. This is a segment of DNA code, with a certain code. And what this promoter does is it attracts and binds to the RNA polymerase. And RNA polymerase, as you remember, is what is the molecule that makes messenger RNA. And these promoters obviously indicate the start of a gene. And once they've found the promoter, then they can sequence that whole part of the gene. And one of the other things you got to remember is they got to look for the introns and the exons parts of the genes. Those are the parts that actually code for part of the gene, and parts that are just kind of DNA that's not really used to code for the gene. Once you have sequenced this, we can start doing things such as gene therapy. And once we know what the sequence are of healthy alleles, what we can try to do is correct genetic defects. And one of the ways they use is one of the tools they use is a virus. And if you remember, a virus can enter a cell very easily, so it becomes an ideal little carrier for a gene. So what they do is they take a virus, they change it so it can't cause disease, and they attach the normal gene to the DNA of the virus. And the virus then, here's the normal gene right here, inserted into the DNA of the virus. They insert the virus into the cell. The virus enters into the nucleus of a cell. The virus then inserts that gene into the DNA of the cell, and then that cell is then placed back into the bone marrow of the patient. This is a short section, and but it's kind of should bring up a couple of small little, not little, but pretty important points about some of the ethical issues with I don't want to say tampering, investigating and changing human DNA. And the whole issue is far too complex to kind of go over in a small podcast, but this cartoon I found what I thought was pretty kind of brings up some of the broader issues about how there are advantages and disadvantages to manipulating human DNA, and that it's not something to be taken lightly.